Hi guys, just wanted to hop on here and give you a quick lecture since we won't be able to meet today. Last time we left off in class, you guys were doing experiment 8.2. So I wanted to go through and explain what they were wanting for that. So the first one was that you're supposed to draw a 1x1 Punnett square to show a cross between two individuals. The information given to us was that there are dominant and recessive traits for PTC tasting and for tongue rolling. So if you look up here, the dominant gene for the tasting is represented by a capital T, and then we have the capital R for the roller. So therefore the little t is for the non-taster and the little r is for the non-roller. So for the first thing in the procedure, it says that we have a man that is homozygous taster roller. So taster roller, homozygous means that they have the same genes for those. So we have a double T, a double big T, and a double big R. Homozygous meaning the same. So he has two of the same dominant genes for both. And then there's a woman who is a non-taster and non-roller. So in order for her to be a non-taster, non-roller, she has to have also homozygous for the recessive genes. So this is why we only need one square, because there's only one possibility here. Okay, so now we're going to do the insides. The only thing that he can give is a dominant T. So he gives a dominant T. The only thing she can give is a little t. So we have heterozygous for tasting. And then we have one R to give here. So we have a heterozygous situation for both. The phenotype, though, would be tasting and rolling, because they did inherit the dominant gene. Okay, now we're going to do the second part. The second part is a dihybrid cross. So it's important that you guys know how to do dihybrid crosses because it's going to be on your test. Okay, so it says that we have a heterozygous taster roller crossed with another of the same genotype. If you look here, this is what the genotype looks like. So we need to figure out what each of their gametes could look like. Okay, we could have this one with that, or this one with the big R. We could also have the big T, little r, and little t, little r. Oops. Okay, so it would be the same for this one because it's the exact same genotype. So we're going to put these across the top and across the side. All right, now we're just going to fill in. We have big T here, big R, and we just fill in across. Remember to put the dominant gene first. Big T, big R, little r. Okay. Just takes a while to fill these out, but it's not hard. We're just filling in what we have. Little t, big R, little r. Okay. These are time consuming, but it's important that you get everything in there so you can figure out all of the percentages, because you're going to be That's part of the test, is you're going to have to tell the percentages. Okay, so the question is, indicate the percentage chance of producing each of the following: roller taster, roller non-taster, non-roller taster, and non-roller non-taster. Okay, so let's circle all of the taster rollers first. Remember, if it has a dominant gene, that means it's going to be a taster, and dominant gene for the R's is going to be roller. So every time you see a big T or a big R, that is taster roller. Okay, we have one here, got one here, and here, here, and that's it. Okay, so we have 16. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16. I don't have it right in front of me, but you would just punch that in the calculator. I think it might be 56.25 or something close to that. So just whatever you get as a fraction, just go ahead and type it into your calculator as a percentage. It should give you a decimal point for the dihybrid cross. Okay, so those are taster rollers, and then we need non-taster roller. So a non-taster would be with the little t, but we would have the big R. So here, and here, and here. I think that should be it. 3 out of 16. Then we should also have 3 out of 16 for a taster non-roller, which is here. 1, 2, 3. And then the last one is the non-taster non-roller, which would be 1/16. Okay, so that's how we do the dihybrid cross. And the percentages, just type in the fraction. Fractions, of course, mean division. So 9 divided by 16, it should give you the percentage, okay? Okay, now we're gonna go over the rest of the chapter. So we left off on page 247, if you wanna look there while we're discussing these. Put a little star by the two terms on page 247, autosomes and sex chromosomes. So just to review, human beings have 23 pairs of chromosomes. All but the last pair are called autosomes. These are all of the chromosomes that code for things that are not determining the sex of the individual. The pair on the end we call the sex chromosomes. These determine the gender or the sex of the individual. So the female would be XX and the male would be XY. Now the Y chromosome doesn't exactly look like a Y. It actually just looks kind of like a half of an X. But we can see how the sex is determined by the gametes given by the mothers and the fathers. So this would be the gametes that the mother could give. So the mother could give 2 different X's. And the father, of course, has XY to give. So if we just do a simple Punnett square, we can see what percentage chance you would have of being a male or female. So you can see 50-50 chance. That doesn't mean that you're going to get, half of your siblings are boys and half of your siblings are girls. It just means that each individual chance has a 50-50 chance. So the significance of this pair, we're going to talk about because there are fewer genes on the Y chromosome because it is a lot shorter than the X chromosome. If there are genes that are only linked to this pair, some of them are only on the X because the Y doesn't have some of the genes. So we call these sex-linked traits. The alleles, some of the alleles are only on the X chromosome. So that means that certain characteristics males will will more likely show than females. Some of the sex-linked traits are color blindness, male pattern baldness. So this is when we say you get it from your mother's side. That's what we mean. Hemophilia and Duchenne muscular dystrophy is also a sex-linked trait, which is where you have your muscles are weak or damaged over time because of this recessive trait. Let's take a look at what this means for, let's say, color blindness. If you know anybody who's colorblind, they're most likely a male. The male color blindness is a sex-linked trait. So let's say you had a mom who was a carrier of the gene. Now, we say that a mother is a carrier when she has a heterozygous situation. So she will not be showing the trait because she has the dominant gene. But if she has the recessive gene on one of her chromosomes, she is a carrier, which means that she can pass it on to her children, and especially to the son who would only have the one X with that recessive allele. Now for these, I'm going to use a different color for the recessive gene so you can kind of trace it through. So let's say we have a mother who is a carrier for color blindness. So she would look like this. Of course, the dominant gene is the big C. Okay, so we have the mother who's a carrier, big C, little c. And then the father is not colorblind. So he's just going to have the dominant gene on his X chromosome, and then of course there's nothing on the Y chromosome because that's it's a sex-linked trait, so it's not on the Y part. So this one would be a female carrier here. This would be a female with both dominant genes, so she would not be a carrier. Both of those females would not be colorblind because, as we learned before, in order to have the colorblind trait or to show it, you have to have both recessive genes. This one, however, will have a male who is colorblind and then a 50% chance of having a male who is not colorblind. So 0% chance of having a female who's colorblind, but 50% chance of having a female who is a carrier, and then a 50% chance of having a male who is colorblind. If you look at example or experiment 8.3, we are going to look at a situation where there is a sex-linked recessive trait for the royal family. So apparently Queen Victoria, is it Queen Victoria? Queen Victoria was a carrier of the recessive sex-linked trait for hemophilia. Now hemophilia is the inability for the body to form blood clots. So you can imagine how this would be, this is a very serious condition. If the person is just even minorly cut, it would be difficult for a wound to heal. They could bleed out, basically. Most people who have hemophilia don't live very long because of that. It just causes all kinds of problems. Queen Victoria was a carrier for hemophilia and actually passed it on to one of her sons. So in this experiment, you're going to do a cross similar to this one. So we'll just switch everything out. Okay, so Queen Victoria was a carrier for hemophilia. We're gonna just mark that with an H. And then her husband did not have hemophilia, so he had the dominant gene. So we have a 50% chance of having a female who is a carrier, and a 50% chance, again, of having a male who has hemophilia. So that is what the experiment looks at here. Now, not every situation with genetics is like black and white, dominant recessive situation. There's all kinds of complications from genes that interact with other genes, genes that switch on and off other genes. We're just going to go over this briefly. If you look on page 250, we have polygenic inheritance, which means that several genes are interacting to produce a trait. For instance, eye color is one of those. I'm not going to go into detail because these aren't on your test. This is just for your information, just to get a little bit more knowledge about genetics. I think height is also probably one of these polygenetic inheritance traits. There's a lot of different genes that are acting together to form whatever trait you have. We have a situation called incomplete dominance, which is a mixing of traits. Now this is most vividly seen in flower colors. So if you have a flower who has a red allele and a white allele, or maybe red is dominant and white is recessive, when you have a heterozygous condition, when you have red and white alleles, it actually shows up as pink. So that's what we call incomplete dominance, meaning that there might be a dominant gene, but when they're together, they actually have a mixing of the two traits. Then we have a term called epistasis, which is one set of alleles affects how another set are expressed. So you might have, this is more like when you have a set of alleles that are the switch for another set of genes. So it might be a switch on or off. Your book uses an example of coat color in mice. So you have a dominant black gene, a recessive brown gene, but you also have this other set of alleles, which is actually a switch to turn those genes on and off. So if the switch is on, then you will have just whatever phenotype of the recessive or dominant genes that we've seen before. However, if those alleles say to switch it off, you will actually have a white coat color. So you have 3 different colors that you could have in the mice, and that's because one set of alleles is interacting with another set. All right, another term is called pleiotropy. Your book actually spelled this wrong. I don't know why. It's not pleiotrophy, it's pleiotropy, which is one gene affects multiple traits. So in the case of something like sickle cell anemia, where the blood vessel or the red blood cells normally are shaped nice and round like a little pancake with a little dent in the middle. In sickle cell anemia, they have sort of a rigid shape, and they might be kind of half moon shaped. So instead of, like just bouncing around in the blood vessels and moving for the other ones, they're not real pliable. They're like real stiff, and they'll cake up together and stack in and form blood clots and different things. So it can lead to all of these different situations because of this one defect. You can have anemia, which is when you have low iron. Iron is something that binds to the outside of the red blood cells. So if you have a defect in the shape of the outside, you're going to have problems with that. Blood clots, pneumonia, pain, heart failure. So all of those things are situations that occur because of this one defect. Now your book talks quite a bit about blood types, which is very interesting, but it's not on the test, so I'm not going to go over that. You can certainly read it on your own. It's very interesting, the way blood types work with genetics. The only other important thing that's going to be on the test are the means by which genetic abnormalities occur. So there are 5 different ways that we can have a genetic disorder. Number 1, and these are on page, well, I think it starts on page 253. So if you want to mark those in your book. Number 1 is autosomal inheritance. Remember that autosome means that all those other chromosomes except for the sex chromosomes. A genetic disorder that is either a recessive or a dominant sort of situation. Lactose intolerant is a recessive autosomal genetic disorder. The next is sex-linked inheritance. These are the sex-linked traits, which we already discussed. Colorblindness, hemophilia, all those things are disorders that we can inherit on these sex chromosomes. Next, we have mutation. A mutation is a radical chemical change in one or more of the alleles. So this might occur as a result of environmental influence or something else going on in the body, something that happens during meiosis. A lot of different things can happen. Number 4, a change in the chromosome structure. You can have a deletion of parts of the chromosome or the genetic material. You can have a gain of it from something. This may occur in meiosis or during mitosis or one of those processes. And the last is a change in the chromosome number. So when you are going through meiosis, instead of the 2 chromosomes splitting up into different gametes, you could have both of them are pulled into one, so that you would have 2 in one and 0 of that particular chromosome set in another one. Usually deletions of chromosomes result in a non-viable embryo. Sometimes we have a situation where you have 2. So when you have 2 in one gamete joining with the 1 from the other parent, you would have 3 instead of 2. This is called a trisomy. Trisomies are actually quite common. Down syndrome is a trisomy situation of the 21st chromosome. So you have an extra chromosome on the 21st set. So sometimes that happens. It usually happens during meiosis. So those are all of the different ways that we can get genetic disorders. Now, I was supposed to hand out stuff for experiment 8.4, which is showing how environmental factors affect genetic phenotype or how genes are expressed. So we'll go over all of that next week, and I'm going to give you 2 weeks to work on that. It's going to take you several days to grow the radishes and to look at the different colors and things like that. We'll talk about that next week. So your assignment for this week is to finish your study guide and check it over with the study guide answers that I sent you. The Module 8 test, which I'm making open book. And if you have any questions, you can always email, text, call me, and I can help you with whatever you need help with. Also the quarterly test, which Mrs. Estep told me that she lets that be open book. And then when you get done with all of that, we still have Module 10 coming up next week. So go ahead and get started on the reading for that and the study guide if you have time. We are going to cover that all in 1 week, so it's best for you to go ahead and get started on that, okay? And I will see you next week.